Okay. Now, Krista, tell me what you are doing in the lab.
So I washed samples and dried them. The samples were washed in chloroform and methanol and dried. Plant samples were just washed with water. And then what I did was the hair samples were then cut with scissors, which wasn't very effective because it was taking up to 40 minutes because I need not only 1 milligram of hair for the carbon and nitrogen, but I need 10 milligrams of hair for the sulfur analysis. So I moved to a different method using an amalgamator, or what's sometimes referred to as a wiggle bug, that actually pulverizes and powders the hair. It's a mortar and pestle type of design. So what it does is there's a stainless steel ball inside here that you put the hair in, you put the ball in, and then you close it up, and it goes into a machine that basically shakes it back and forth at 3,200 revolutions per minute. And the fur then changes from going to something that looks like that, which is very frustrating, to, let me pick a good sample here, powdered. And this is approximately the same amount of hair. It works great for the moose hair and the caribou hair, which tends to stick to everything, and it powders it down to a fine pellet. And then what happens is that 1 milligram of that gets put into a tiny little tin cup, and it gets folded. Now it's these ones here that are done. So let me move the bills here and put this over so you can see that inside here, actually let me put it right down on you can see that there's tiny little tin cups that have been rolled and filled and measured and then rolled up, and they will be sent.
So inside the tin there is some samples, right?
Yeah, there's 1 milligram of hair. These are the hair samples. So
for plants? And
the plants, it's the same. With the plants, I just used a mortar and pestle, a stainless steel mortar and pestle that I could wash in between, and so that I wouldn't get any cross-contamination, but the method's the same one. So the plant samples, they've been divided up into the lichen, and there's different types of lichen, so there's 30 lichen samples there, and 20 spruce and 6 pine,
So, and the purpose of this thing is that you will look at diet overlap between a number of predators and plant,
yeah,
also including predators of predators and prey, also
yeah,
predators of plant.
yeah.
And the only missing species in the ecosystem is bears now?
Yes, the black bears and grizzly bears. I have 1 black bear sample, and I'm getting a number of grizzly bears, I have 1 grizzly bear sample, and I have a number of black bear samples, but the black bears are from Manitoba. I don't have.
And should someone with initial GS help you, substantially we are open to collaboration in this study of diet overlap as it relates to landscape variables, so it's also including human development, right?
That would be fantastic. And the reason that I've moved away from scissors to this powdering method is that there's going to be 500, probably close to 1,000 samples. I've got 500 washed and dried right now.
So what is the total number of samples that you have now?
Right now that I'm sending to California for hair? In total, I have right now 500. 500 samples and broken
from Manitoba and
Alberta combined. Yeah.
Okay, and all the species in the ecosystem, basically.
Yeah, except for the bears.
Okay.
Yeah, and beaver. Beaver in Alberta is my big problem too.
It's okay. It's okay. I don't think it's significant in diet of predators. Thank you.